Your blood type looks like a hospital label, but one global pattern refuses to fit the migration map. A nurse writes A positive, O negative, B positive, or AB negative on a chart, and it feels like the plainest fact in the room. It tells a blood bank what can be transfused. It tells a doctor what has to be checked before surgery. For most people, that is where the story stops. But when scientists compared blood types across populations, those labels started acting like migration signals. ABO sits on chromosome 9. Rh sits on chromosome 1. They are inherited markers, passed through families long before passports, borders, or surnames existed. The broad map almost made sense. Type O became strikingly common across much of the Americas. Type B rose across parts of Asia and northern Eurasia. Type A appeared often in parts of Europe and the Near East. Then, Rh negative broke the smooth picture. It was rare across much of the world. Then, in Western Eurasia, it formed a concentration that did not fade the way a simple migration wave should fade. That is the pattern that does not fit cleanly. To understand why it matters, the blood label has to be separated from the myths around it. Blood type is not a race. It is not an identity certificate. It is not a measure of intelligence, strength, value, purity, or destiny. No blood group makes a person older, better, more original, or more human than anyone else. The caveat matters here. ABO describes molecules called antigens on the surface of red blood cells. If your cells carry the A antigen, the chart may say A. If they carry the B antigen, it may say B. If they carry both, it may say AB. If they carry neither, it may say O. The main instructions for that system are controlled by variants in the ABO gene on chromosome 9. Rh is a different system. The familiar positive or negative label usually refers to the D antigen, one of the most important parts of the Rh blood group system. If that D antigen is present, the chart says positive. If it is absent, the chart says negative. The major gene behind that difference, Rhd, sits on chromosome 1. So a blood type is two systems placed together on one form. ABO tells one inherited story. Rh tells another. Hospitals put them side by side because medicine needs them side by side. Population geneticists looked at them for another reason. Before modern DNA sequencing, blood groups were among the easiest inherited markers to measure. A drop of blood could show a pattern in a family, a village, an island, or a whole region. By the early 20th century, after Carl Landsteiner identified the ABO system in 1900, researchers began turning blood groups into maps. Later, DNA-era studies showed those old maps were crude, but not useless. They could reflect migration. They could reflect founder effects, when a small group gives rise to many descendants. They could reflect isolation, when people marry mostly within a region. They could reflect bottlenecks, when war, famine, disease, climate, or conquest cuts a population down so sharply that only some lineages pass through. One marker can never tell the whole story, but one marker can keep a trace. Once that is clear, the world map becomes more than a medical chart. Imagine a lab screen filled with red blood cells, then a map shaded by frequency. The colors do not stop at borders. They flow, blur, thicken, and disappear. They look less like modern countries than old routes of movement. Type O is the first broad signal. It is found across the world, but in many indigenous populations of the Americas, it became especially frequent. That pattern has drawn attention for more than a century because it looks like a genetic echo of a difficult passage into a new continent. The old migration map points toward Northeast Asia, Beringia, and the peopling of the Americas. Beringia was the land and coastal region that once connected Siberia and Alaska during lower sea levels. The people who crossed or moved along that world did not carry every possible genetic variant in equal amounts. They carried the variants present in the smaller ancestral groups that survived the route. That is, a founder effect. A small number of ancestors can leave a large signature if their descendants expand into a vast new space. If type O was common among those founding groups, later generations could make it look even more dominant. Then came the second pressure. Many indigenous populations of the Americas passed through catastrophic bottlenecks after European contact. Epidemic disease, warfare, enslavement, displacement, and social collapse did not strike evenly. Entire communities disappeared. Surviving groups could carry a distorted version of an older map. That does not mean type O belongs to one people. It means the frequency of a blood type can be shaped by ancient migration and later historical damage at the same time. The genome kept one more record. Type B forms a different pattern. It tends to appear more often across parts of Central Asia, northern and eastern Eurasia, and parts of the Indian subcontinent than in much of Western Europe. The map is not a single arrow. It is a broad zone shaped by old population movements, steppe corridors, trade, marriage patterns, and regional histories far older than modern names. Type A creates another layer. It is common in many European and Near Eastern populations, with important pockets beyond them. Some researchers have discussed it alongside farming expansions and population growth in Western Eurasia, but the relationship is not simple. Farmers moved, hunter-gatherers survived. Later steppe groups entered Europe. Communities mixed, split, and mixed again. So the ABO map can tempt people into easy stories. O means this continent. B means that region. A means that migration. But the closer the map is studied, the less it behaves like a set of hard walls. These are gradients, not borders. Still, the ABO system mostly behaves like population history. It rises and falls across geography. It can be influenced by migration, founder effects, isolation, selection, and later bottlenecks. The pattern may be uneven, but the rules are familiar. Then the RH map appears. On the same shaded world map, RH negative does not look like a gentle worldwide gradient. It is uncommon in much of East Asia. It is rare among many indigenous American populations. It is also uncommon across many African populations, though the RH system in Africa is genetically diverse and cannot be reduced to one simple European pattern. Then Western Eurasia stands out. The anomaly is not that Rh negative exists there. The anomaly is the concentration. In parts of Europe, and especially in historical attention around Basques and nearby Western European groups, Rh negative appears at much higher frequencies than most of the world. For decades, that difference pulled in speculation far beyond the evidence. Some people turned it into mythology. Some turned it into claims about hidden races, alien origins, or secret bloodlines. Those claims are not science. They are stories, built around a real pattern, then cut loose from the evidence. The evidence points in a narrower direction. The RHD gene on chromosome 1 controls much of the familiar Rh positive and Rh negative label. In many Europeans who are Rh negative, the common mechanism is the deletion, or functional absence, of the RHD gene. Without the D antigen on the red blood cell, the person is classified as Rh negative. That mechanism matters because it makes the pattern biological, not symbolic. There is no mystical quality in the blood. There is a gene region, an antigen, an inherited variant, and a population history that changed how often that variant appeared. If Rh negative were only a simple, ancient marker spreading evenly from one origin, the map would be expected to fade more smoothly with distance. A center would have edges. The pattern would thin out gradually as populations mixed and moved. Instead, the map is sharper. It forms a Western Eurasian concentration that does not behave like a simple global wave. It looks compressed, as if older population structure, isolation, and later expansion pushed one marker into a regional shape. That is where the old map failed. The Basques became a focus because they were already unusual in the European story. Their language is not Indo-European. Their homeland sits in the Western Pyrenees between modern Spain and France. Their history seemed to preserve something older than the surrounding linguistic map. When high Rh negative frequencies were discussed in that region, the temptation was obvious. Maybe the blood type came from a lost people. Maybe it was proof of a hidden origin. Maybe one isolated community carried a living record from before the rest of Europe changed. There is a responsible version of that question, and there is a reckless version. The responsible version asks how founder effects, drift, and regional isolation could raise the frequency of Rh negative in some Western Eurasian populations. Drift is the random rise or fall of a variant across generations, especially in smaller populations. A variant does not need to be better to become common. It only needs the right history of births, deaths, isolation, and chance. The reckless version turns one blood label into a fantasy of purity. The reckless version has to be rejected. Rh negative is not proof of a superior lineage. It is not proof of an untouched tribe. It is not proof that one population stands outside humanity. It is a marker whose frequency was shaped by population history, and population history is always messier than a slogan. That would make the pattern a regional curiosity, except the mechanism points deeper than a single tribe or modern nation. The deeper layer begins with why people reached for extinct humans in the first place. Once Neanderthal DNA entered the public imagination, every strange genetic pattern started attracting the same question. Could this blood type come from Neanderthals? Could Rh negative be a trace of a vanished human group? Could one label on a hospital form be the last sign of an ancient species? The question sounds cinematic because extinct humans really did leave DNA in living people. In 2010, researchers at the Max Planck Institute and other teams showed that many people outside Africa carry Neanderthal ancestry. Denisovan ancestry was identified from a small bone fragment found in Denisova Cave in Siberia, then traced most strongly in parts of Oceania and nearby regions through genome-wide comparison. That method is the key. Neanderthal and Denisovan signals are not detected from one blood label. They are detected by comparing hundreds of thousands, then millions of positions across the genome. Scientists look for long patterns of shared mutations, not a single letter on a hospital chart. Rh negative is not a Neanderthal badge. It is not a Denisovan marker. No responsible genetic model treats it as proof of descent from an extinct human species. The first model broke there. The real story is less theatrical, but more human. Western Eurasia was not a stable stage where one ancient population simply became modern Europeans. It was a harsh, shifting corridor between ice, steppe, forest, coast, and mountain refuge. During the Upper Paleolithic, people lived through repeated climate swings. Ice sheets expanded, habitable zones contracted. Some groups survived in refuges, places where climate and geography allowed people, animals, and plants to persist, while other regions became nearly impossible to inhabit. A refuge can change genetics without creating a new species. If a small population survives in a protected region, its genetic variants may become more common by chance. When the climate improves and descendants expand, those variants can spread outward with them. This is how ordinary inheritance can preserve the shape of ancient survival. Ancient DNA from Upper Paleolithic and Mesolithic Europe has shown that the continent was repeatedly reshaped. Skeletons from caves, rock shelters, and burial sites do not point to one continuous line moving peacefully through time. They show replacement, mixture, disappearance, and return. Some hunter-gatherer groups contributed strongly to later populations. Others nearly vanished from the genetic record. Then, farming populations moved into Europe from Anatolia and the Near East. Later, steppe pastoralists brought another major ancestry layer. Each expansion did more than add people. It changed frequencies. A variant that had been common in one surviving pocket could be diluted in one region, preserved in another, and amplified somewhere else. The second layer was stranger. Rh negative may have become concentrated through this kind of layered history. Not because it marked one pure people, not because it came from a lost species, because Western Eurasian populations passed through ancient structure, local isolation, founder events, drift, and later expansions after bottlenecks. A bottleneck is a narrowing. Imagine a wide river forced through a stone channel. Only part of the water passes at once. In genetics, only part of the variation passes through a crisis. The descendants on the other side may carry a pattern that looks intentional, even when history was driven by survival and chance. That is why a blood map can look mysterious without being supernatural. The Rh negative concentration is a surviving distortion. It is what can happen when small communities endure climate pressure, remain partly isolated, then contribute unevenly to later populations. The marker becomes a crease in the map. It shows where the past folded. This also explains why the Basque discussion became powerful but incomplete. The Basques may preserve important regional history, and their genetic profile has helped researchers think about continuity and change in Western Europe. But no living population is a sealed container from the Ice Age. Every group has history before it, contact around it, and change within it. The anomaly is larger than one community. It belongs to the broader story of Western Eurasia, where old refuges, mountains, coasts, farming expansions, and later migrations left a blood type pattern that still looks uneven today. The first answer was not an extinct species. The stranger answer is that ordinary inheritance can preserve the shape of ancient survival. Now return to the modern form. A person sees O negative before surgery and thinks about transfusion. Someone else sees A positive on a pregnancy record. Another sees B positive on a blood donor card. Another sees AB negative and remembers being told it was rare. In daily life, those labels are practical. They belong to hospitals, blood banks, and emergency rooms. At the level of one individual, they prove very little about ancestry. An O negative person is not automatically from one region. A B positive person is not automatically from Asia. An A positive person is not automatically European. An AB negative person is not carrying a secret identity. One marker is too small. At population scale, though, thousands of individual labels can form a pattern. Type O frequencies in many indigenous American populations can point toward founder effects and severe later bottlenecks. Type B gradients across parts of Asia and Eurasia can reflect old zones of movement and population structure. Type A distributions in Europe and nearby regions can be discussed alongside farming-era expansions, later mixtures, and local histories, while still remaining debated and complex. Rh negative supplies the sharper anomaly. It is rare across much of the world, yet concentrated in Western Eurasia in a way that does not reduce to a simple global fade. That is why the title matters. The blood type map is not an ancestry test in the modern sense. Genome-wide DNA has replaced blood groups as the stronger tool because it can compare vast stretches of inherited material. It can detect ancestry components, ancient mixture, cousin relationships, population clusters, and archaic introgression with far more power than ABO or Rh. But the old blood maps still matter. They were among the first biological maps to show that human history was uneven. They showed that continents were not smooth categories. They showed that isolation could make one village different from the valley beside it. They showed that a tiny inherited marker could remember an ancient narrowing that no written document recorded. There is also a harder ethical pressure. Modern frequency maps can be damaged archives. Colonization changed them. Epidemic collapse changed them. Forced displacement changed them. Sampling gaps changed them. Some communities were studied heavily because outsiders found them interesting. Others were ignored, mislabeled, or treated as footnotes. When a map shows type O dominance in parts of the Americas, it is not only showing ancient founder effects. It may also carry the shadow of population collapse after contact. When a map shows low or high frequency in an isolated community, it may be showing survival under pressure, not a romantic symbol of untouched ancestry. The evidence has to be read carefully. Disease may also have shaped ABO variation in some places. Researchers have studied links between blood groups and susceptibility to certain infections, and some hypotheses suggest that natural selection may have influenced frequencies over long periods. But this documentary is not medical advice. No viewer should infer personal risk, treatment, protection, or health destiny from a blood type discussion. A population pattern is not a personal diagnosis. The old blood maps sit between medicine and ancestry, and that makes them easy to misuse. They are close enough to the body to feel personal. They are broad enough to tempt people into identity claims. They are ancient enough to attract myths. But the correct reading is narrower and stronger. Blood types can suggest population history when studied across many people and compared with archaeology, migration models, ancient DNA, and genome-wide data. They cannot tell one person who they really are. They cannot rank groups. They cannot assign belonging. They cannot turn biology into a border. The title's anomaly is not that one blood type is magical. The anomaly is that one inherited system kept a distorted trace of movement, isolation, chance, and survival. That makes the ordinary hospital label look different. ABO gives broad continental echoes. It records founder effects, old movements, and regional histories through a system on chromosome 9. O becomes a powerful signal in many parts of the Americas, partly because of founding populations and later bottlenecks. B rises in broad zones across Asia and Eurasia. A appears often in parts of Europe and the Near East, with a story complicated by farming, migration, and mixture. Rh adds a second map from chromosome 1. It asks a sharper question. Why should Rh negative remain rare across so much of the world, yet reach much higher frequencies in Western Eurasia? The answer is not one tribe. It is not one nation. It is not one fossil species. It is not a secret caste of blood. The final model is more demanding. Blood types are small genetic signals shaped by movement, isolation, selection, drift, and bottlenecks. They are real but incomplete. They are inherited but not destiny. They can carry ancestry echoes, but they cannot replace the whole genome. Genome-wide DNA changed the field because it gave scientists a much larger archive. Instead of reading 2 labels on a hospital form, researchers could compare entire chromosomes. Instead of guessing from blood frequencies alone, they could test ancient skeletons, living populations, and extinct human DNA against one another. Yet the old maps still have a place. They show what researchers noticed before sequencing made the genome visible. They show that human history was never a clean tree with neat branches. It was a record of crossings, dead ends, expansions, refuges, and returns. It was a map where some lines faded smoothly and one pattern did not. The hospital label returns at the end as something both smaller and larger than it first appeared. Smaller because it cannot define you. Larger because it belongs to a chain of inheritance that began long before the form, the clinic, the nation, or the family name. A blood bag, a family tree, and a migration map seem like separate objects. One hangs in a hospital refrigerator. One sits in a drawer with old photographs. One appears on a classroom wall with arrows moving across continents. But in the genome, those objects overlap. The pattern that did not fit was not a ranking. It was not a purity claim. It was not proof of better blood, older blood, or a hidden human caste. It was evidence that Ancestry moved through bottlenecks, coastlines, refuges, epidemics, isolated communities, and chance. That is, the record blood types can still preserve. If you want careful genetic history, ancient DNA mysteries, and the evidence most people were never taught to read, subscribe to the Genome Archive. The human story was never written in one blood type, one continent, or one line of descent. It was written through mixture, survival, and memory.